Hello students. So this is second part on anaerobic bacteria where we are just going to discuss about the culture techniques which are used for anaerobic bacteria. Okay. In first part we had already discussed about the basics, some characteristics, examples and which are the media which are widely used for anaerobic culturing. Okay. So before starting with techniques, just let's revise what are the characteristics of anaerobic bacteria. Okay, so anaerobic bacteria they are unable to grow in atmosphere where oxygen is present. They use fermentative pathway in which organic compounds they serve as final electron acceptor. Okay, not oxygen. Then they are killed when they are exposed in atmosphere for say minimum of 10 minutes also. So some anaerobic bacteria or some anaerobes they are tolerant to small amount of oxygen while some are facultative as they can grow with and without oxygen. Okay, so these are the basics. So now about the cultivation of anaerobic bacteria. So obligate anaerobes are strict anaerobes which will only grow in the presence of zero oxygen. Okay, when all the oxygen from the given environment is excluded, that is, it is removed from the that particular environment, or we can say if even if the media. Okay, so as the media is deprived of oxygen, the obligate anaerobes they will grow happily. Okay, otherwise they will not grow. So such an environment is very necessary to be established, and we can do that by using one of the following methods. So first is pre-reduced media, then anaerobic jar, gas pack, anaerobic chamber, gas pack, anaerobic jar, candle jar and biological method. So of which I think you must be familiar with anaerobic jar and a candle jar. Okay, as these are widely used in colleges. Okay, and if you want to see or visit and get to know more about anaerobic chamber, and if you don't have that facility in your institute or college, then just ask your professor to arrange a visit where a lot of work is being carried out on anaerobes and where anaerobic chamber is present, so how it works, and or at least have a demonstration of anaerobic chamber working. Okay, so ask for that in your college to your professors. Then about first technique, that is pre-reduced media. So we use this to cultivate anaerobic culture. And how? So first is about the pre-reduced media. So these are the media which or medium which are commercially available, but you can also prepare them in your lab. So it depends on you what is your priority. Okay, or with what you can work with, that it's up to you. So you can prepare in your lab, okay, and try to flush your media with lots of nitrogen free oxygen free nitrogen to get the oxygen reduced or drive off. Okay, or even oxygen free carbon dioxide, so that the concentration of carbon dioxide is increased in your medium. Okay, so it depends on you what is the best thing works for you. You can use that. So about preparation of pre-reduced media, the culture media is boiled for several minutes to drive off the dissolved oxygen, and a reducing agent, that is cysteine, is added for further lowering the oxygen content in your medium. Now oxygen free nitrogen is bubbled through the media to keep it anaerobic. Okay, so oxygen free nitrogen gas is bubbled, that is what I am calling here flushing your media. So then media is then dispensed in tubes which is being flushed again with oxygen free nitrogen. It is then stoppered, means capped tightly, and sterilized by autoclaving. So such tubes can be stored for many months before using. So during inoculation, these tubes are continuously, you have to again flush them with oxygen free CO2. Okay, so there is a way, that is using a cannula, that is gas cannula is, you can say, or you can imagine it like a small gas tube. Okay, which you can use in your media, and you can flush your media and then again restopper it and incubate. Okay, so the procedure of using a pre-reduced media is you take a tube which contains atmosphere of oxygen free nitrogen. Okay, that is oxygen free media. Okay, and that is why it is named as pre-reduced. So to inoculate, you remove the stopper. Again, you have to insert a gas cannula, flush the tube continuously with oxygen-free carbon dioxide and maintain the anaerobic conditions. So now you inoculate the media and then you can incubate it after restoppering. Okay, just a simple way. But here you have to keep in mind that flushing your media with oxygen-free CO2 and N2 is very important to maintain the anaerobic conditions. Now about anaerobic chamber. So in second slide you will have that image where you can refer to whatever I am going to explain now. Okay, so actually it is a plastic anaerobic glove box, okay, which contains atmosphere of H2, CO2 and N2 but no O2, that is there is no oxygen. What all is present in the atmosphere? Hydrogen, carbon dioxide and nitrogen. So culture media are placed within a chamber by means of airlock, which is evacuated and refilled with N2. So you cannot just open your anaerobic glove box and or anaerobic chamber and keep your plates inside and then close the door and start working. No. So you have to use the airlock. So what is the airlock? The purpose of airlock is to remove the oxygen that is present in air. So the oxygen is removed, evacuated, and the airlock is filled with the nitrogen. Okay, and once that is done, then you can open the second door of your airlock and take your plates inside or keep your plates inside your anaerobic chamber. So this is done to maintain the atmosphere of zero oxygen. Okay, so from the airlock the media is placed within the main chamber. Any CO2 in the media is slowly removed by the reaction with H2 by forming water, and this is aided by the palladium catalyst. So palladium catalyst is very important as it works with the oxygen and hydrogen to form water, and it helps in removing the remaining uh, concentration of your oxygen. Okay, so after being rendered oxygen free, the media is inoculated within the chamber, okay, by using or by the means of glove port, and again it is incubated in the chamber itself. You cannot now or you cannot take your plates out of your anaerobic chamber and keep it in your aerobic regular chambers. Okay, you have to incubate anaerobic plates in anaerobic environment, so you will be incubating them in your anaerobic chamber itself. So now for working, there are two ports, glove ports. Okay, so it has glove ports and rubber gloves attached to it so that you can operate easily. Hmm? So this is the anaerobic chamber, which is made up of plastic. So this is the airlock. There are some gas tanks placed nearby so that the required atmosphere can be generated by using hydrogen, carbon dioxide and nitrogen. So from airlock, after evacuation of oxygen and refilling the airlock with nitrogen, the plates are kept inside your anaerobic chamber and you can work there easily from the glove ports. Okay, after inoculation, you have to incubate in your anaerobic chamber itself, right? So the procedure is given here, you can read it. So next we will move towards is gas pack. So gas pack is generally used in place of candle jar. So the packet consists of a bag containing Petri plate and CO2 gas generator. The gas generator is crushed to mix the chemical. Okay, you can see here there is a gas generator which is crushed to mix the chemicals which it contains to start the reaction of production of carbon dioxide. The gas reduces the concentration of oxygen in the bag by about 5% and provides CO2 concentration by about 10%. So you can say comparatively the carbon dioxide environment is more in your um, gas pack. So that is the reason, or gas generator is the reason which that is why we use it while you are working with anaerobic bacterial cultures, is to generate the CO2 concentration, more CO2 concentration, and you have to evacuate, you have to remove the oxygen concentration. Okay, so the media is inoculated and it is placed in the jar. Then water is added to the gas pack generator envelope, causing the evolution of CO2 and H2. The H2 will now react with the oxygen that is present on the surface of palladium catalyst and it will form water. Okay, and this establishes the anaerobic condition. The CO2 aids the growth of fastidious anaerobes which sometimes fail to grow. Now, the more concentration of CO2 which is generated by the gas pack, it will aid the growth of fastidious anaerobe, okay, strict anaerobes, which sometimes fail to grow or they grow poorly in its absence. Okay, so this CO2 concentration, high CO2 concentration, will help the growth of anaerobes. An anaerobic indicator strip is also present, okay, which has methylene blue solution on it, and it is working as an indicator which indicates that the environment is now deprived of oxygen. Okay, so the color changes from blue to colorless when the level of concentration of oxygen gets reduced. Next is anaerobic jar. So this, I guess you have seen this in your institute. Okay, if not, then just ask your professor to have a simple demonstration. Now anaerobic jar is heavily walled jar with a gas tight seal within which tubes, plates or other containers to be incubated are placed along with the H2 and CO2 generating system. So now here also you can see there is a gas pack system that is having some chemicals. You will see or you will read that in next slide. So here when the reaction starts, the combination of the chemicals will start evolution of CO2 and H2O. The H2O will now react with the palladium catalyst where the oxygen is present and it will start generating water. Okay, so again the same principle of palladium catalyst is been applied here. So non-stringent anaerobes can be cultured within anaerobic jar. Okay, non-stringent anaerobes means non-obligate anaerobes, they can be cultured. So inoculate the media and place it in the jar along with CO2 and H2 generating system. After the jar is sealed, the oxygen present in the atmosphere within the jar as well as that dissolved in the culture media is gradually used up through the reaction by hydrogen in the presence of catalyst. Okay, as I explained. So this is what all the components of anaerobic jar are, that is gas pack anaerobic system with inoculated petri plates, gas pack generator envelope, anaerobic indicator strip, okay, and the catalyst. So the gas pack generator envelope has sodium bicarbonate and sodium borohydride, okay, and they are mixed with small amount of water to produce CO2 and H2O within the anaerobic jar. The palladium catalyst is used in the jar which combines O2 with H2O to remove the water. Okay, remove the oxygen by forming water. Then next is about candle jar, very basic experiment of oxygen or burning. Oxygen supports burning. That principle is getting applied here. So many microphiles are also capnophiles as they require the elevated concentration of carbon dioxide. Okay, so many microphiles are capnophiles, which means they require high concentrations of carbon dioxide to grow. So they can be easily cultivated in laboratory by using a candle jar. A candle jar is a container into which a lit candle is introduced before sealing the container's airtight lid. The candle's flame burns until extinguished by oxygen deprivation, which generates the carbon dioxide rich environment and oxygen poor atmosphere in the jar. So many labs also have access directly to carbon dioxide and can add the desired carbon dioxide levels directly to the incubator as if they want to grow the microaerophils. Okay, so if that particular facility you have, then you can use and you can directly uh, introduce the carbon dioxide into your incubators, okay, and maintain the levels. Candle jar are used to grow bacteria requiring an increased CO2 concentration, and candle jar increases CO2 concentration and still have some O2 of aerobic, leaves some O2 for aerobic capnophiles. Okay, so candle jar increases the CO2 concentration, but there is no guarantee that you will get zero concentration of oxygen. Okay, you will not get rid of all the oxygen present in the jar. Okay, say for example the oxygen from the media will not be get um, removed, right? So that is the oxygen which is present and that can be used up by aerobic capnophils. So that was the another one. And lastly about biological method. So biological method is simple. Take a plate, media plate, divide it into half. So on the first one half side, you will stick a known aerobic bacteria, for example, Serratia marcescens, and on second half you can uh, inoculate or stick anaerobic sample that is to be tested, or you have a known anaerobic culture, you can use that. Okay, so on one half you have anaerobic sample, on one half you have aerobic sample. So what will have aerobic sample or aerobic bacteria will consume all the oxygen present inside the petri plate, and it will create anaerobic condition for supporting the growth of anaerobic sample. Okay, so how this is maintained, as your petri dish is sealed with wax or paraffin and it is cultured in aerobic environment. Okay, so even though you are culturing or incubating it here in aerobic environment, what will happen, your plate will not get exposed to oxygen as you are sealing it with wax or paraffin. Okay, so once the aerobic bacteria grows well, all the oxygen is consumed, the anaerobic conditions are maintained, and your anaerobic culture can grow now. Okay, so these are all the culture techniques which are widely used for anaerobic culturing. Okay, so I hope this video helps you out. Thank you for watching. Do like my videos, do share my videos with your friends, and do subscribe to my channel. Thank you.